So, good afternoon, everyone. The previous class, we sorry, can you hear me? Hello.
Yes, sir. Yes, sir.
Okay. So, in the previous class, we have discussed about the we overall introduction. We go through with the classic definition of nanotechnology. What is nanotechnology? How properties of material changes from macro to nanoscale, what is the macro scale, what is the nanoscale, how it matters to be in the nanoscale, the uniqueness of the nanoparticles, their dimensions, their 0D, 1D, 2D, 3D dimensions, their classifications and all. And we also started talking about the target of this course, nanoscale modeling and simulation, which I will still continue today because this is the fundamental, and you all have to understand where we are going, because if you don't understand what is the purpose of the course, it is very difficult to understand what we are going to learn in the course. Then we are definitely talking about something which is a merger of these two fields, nanotechnology and biotechnology, which is nanobiotechnology. So let's come to again our course, what we are going to study. I've seen this slide, nanoscale modeling and simulation. I talked about there are three parts: nanoscale, modeling, simulation. So we asked you questions. What do you mean by modeling? What do you mean by nanoscale material modeling? And how are you going to perform modeling of nanoscale material? I didn't receive any answer from you guys. I think there is a miscommunication. So make it clear. You guys write the answer of this question and mail it to me. Is it clear to all of you now?
Yes, sir.
Yes, sir. Yes, sir.
Okay. But I would love to hear if any of you want to talk about this. What do you mean by modeling? Anyone? No idea. You don't understand anything when you hear the term modeling? Somebody is trying to say something, but somehow the sound is not clear.
Sir, organizing something for any specific purpose.
Okay. Fair enough. Any example?
So to perform concepts in a laboratory scale, like real world concepts.
Rutuja, Pradeep, both. Remember the rule, because we cannot see anyone. I have my whole slide and I can only see myself in the video. So it would be great if you just kindly spell your name like Rutuja and then talk. Then it would be really good for everyone. Okay. Yes, continue, please.
Yes, sir, Rutuja.
Yep, Rutuja, please.
So to perform the real world concepts on a laboratory scale, just to find out how the process takes place and what could be the outcomes.
So what you mean is there is a real entity. We want to perform experiment on that, and that's why we create something which represent that, and I call it a model, right?
Yes, sir.
Fair. Anyone else? Okay. So Pradeep and Rutuja have tried their best to define. They have their perspectives, and I will answer in my way. But before that, we will go to the next questions. Like, if that is modeling, what do we mean by nanoscale material modeling? And how we are going to perform nanoscale material modeling? Again, anybody? Like, you say modeling is creating an entity so that we could perform experiment on that. So how you think that would be done in case of nanomaterials? And it's a little bit of unique situation, because if you see, by describing a nanomaterial is taking the smallest fraction going into the atomic level. So what less than that? What we are, kind of develop when our entity is already smallest. Are you getting me?
Yes, sir.
So anyone? What you mean modeling with nanoparticles? Nobody? So I have redesigned the question, and because you guys don't understand or don't connect the fact that how the modeling would be done in nanoscale, I think that is because we should answer many fundamental questions in the field of nanoscience, nanoscale. So I have listed a few questions. First one, what do we mean by nanoscale? I discussed it in the last class, but again, I want you to answer it. So what do we mean by nanoscale? What is the nanoscale used for? Going into that nanoscale, what would be the application? What is nanoscale structure? What are the dimensions, classifications when we deal with the nanoscale? What is nanoscale range? What are nanoscale particles? How do you find nanoscale when you are searching, when you find that you are on the nanoscale point? How nanoscale can be visualized? How can we see the nanometers? Kind of like similar questions. And what do you mean by nanostructures? So I put 9 questions, and I think how many of you are here as a student? 9. So if you allow me, I could assign them for you. You all like use 1 or 2 slides to describe those questions. So for A, can you say your names, please?
Or Asmita here.
Asmita. Okay. So you don't have to say now. Okay. You go home, you read about that, and you use 2 slides to explain. Okay. And give a small 1-page write-up. Okay. You could use figures and all to describe. Okay, Asmita.
Okay, sir.
Next, B. Do it quickly. Don't, waste time so that we have to go to the next segment. Rutuja. Next. See?
Yes, sir.
Hmm?
Yes, sir.
No, I'm not including. Are you guys taking this class or auditing this class? Ria.
I'm taking this class.
Oh, you guys are like you are enrolled in the class?
Yes, sir. Yes, sir.
Oh, I didn't know that. Okay, then I need more questions. Next. Guys, you don't need marks and all this immaterial, right?
Advaita.
Guys, you don't need marks because this will carry marks. This is not just for fun. We are not here assembled to do some fun so that only the girls are coming and joining here.
Sir, Tejas Man.
ठीक है spelling?
TEJASH.
अच्छा.
TEJASHMAN.
हेलो सर. ये सर ज्योति सर.
Next.
Sir, Akanksha.
Spelling ठीक है?
No, sir. Actually, it's double A.
ये एक्स्ट्रा वाला जो इफेक्ट मुझे नहीं आता. हाँ, कहाँ?
Sir, initially. In the start.
Initially.
So, yeah, double A. Okay, sir.
Next. Next.
Shashi.
Sasi. SASHI.
SHI.
SHI. Correct. Next. I. Next. बहुत सारे लोग बाकी है अभी. कोई तो बताओ. Hello. I think Pradeep, as I know his name. So this is the assignment for now. Rest of the student will come with the next questions. Okay. What you have to do? 2-page PPT with 1-page writing. Okay. Clear to everyone?
Yes, sir.
Yes, sir.
Good. So now we are going to the part modeling. We try to understand nanoscale. Nanoscale is the basic. Everyone in the course will enlighten you about nanoscale, nanotechnology, and all these things. Modeling is what exclusive of this our course. So can we model nanostructure? Before going to this, I bring you another question with very different side. What do we mean by modeling something? As we are discussing, as Rutuja, Pradeep have already talked about, it's something you want to replace so that you could work on it. More elaborately, modeling have parts, like 2 categories. One we call design, another we call develop. What is modeling? Designing? Designing is change or modify according to a requirement. What that means? Means you have something, you don't have to exactly develop it, exactly form it. but you want to change or modify according to requirement. For example, I am here. I am a faculty of BSB, Bioscience and Biotechnology and Nanotechnology. I don't have to reform myself, but I have to take preparation according to presenting my things to you. So what I have to model for representing you is to design the material according to the course. So my modeling of the course is not developing a new thing, designing things from the existing knowledge. Develop is building something from the scratch. You don't have something, you really like take the smaller units and then you develop the model. And if you understand this difference, in this point, I would like to introduce you about the biological system, because in the true sense of modeling is developing. Whereas when we deal with nanoparticles are the smallest volume, smallest structure. We generally apply design, which we are going to do and going to describe about. But I also want to show you how we could develop a model. And so I'm introducing the biological system. Also, you assemble the subunits into meaningful structure is development of a model. As I told, I want to introduce biological system, and biological system is very interestingly jail with the field nanotechnology. So as we define nanotechnology involving the research and technology development at the atomic, molecular or macromolecular level in the length scale of approximately 1 to 100 nanometer range. Biotechnology is the use of biological process, organisms or system to manufacture products intended to improve the quality of human life. Now, the interface of these two worlds, this is called nanobiotechnology, a very modern and current field. It uses nanotechnology to analyze and create biological nanosystems. It uses biological materials and structure plans to produce technical, functional nanosystems. Before going to, I should talk about the components. When you look at life, the life is rather defined as the central dogma. What is central dogma? In the life, three important macromolecules exist without which life cannot proceed. They are called DNA, mRNA, and protein. Now, DNA creates a new DNA by itself. It's a auto process, the process, biological process called replication. So DNA synthesized or model a DNA from by the process of replication. DNA form mRNA or messenger RNA by the process of transcription, and mRNA develop protein by the process of translation. Both DNA and RNA are similar type of molecule. They are called polymer of nucleic acid. And protein is the polymer of amino acid. I know many of you lack fundamentals here, so I would like to go back to give you a 101 learning about biological macromolecules, because once we will know about the biological macromolecules, it would be easy for you to understand how the knowledge of biological macromolecules would be utilized towards development of a model. And once we develop a model, we will come to, so you will learn the development of a model, you will see a real modeling, and then we will come to designing of a modeling, which is modeling of a nanoparticle depending on the requirement of a target. So DNA as a nanomaterial, we have already talked about. If you look at one turn, the DNA is 3.4 nanometer. Between two bases, this is 0.34 nanometer. So it seems like DNA is kind of a zero D nanomaterial, like a nanoparticle, but it's actually not, because if you look at the chromosome, and I will talk about this little elaborated again, and you will understand why I'm going to talk about that. So when you look at chromosome is the, in the living body, it is the unit is a cell. Okay, like if you look at a home, a building, the unit is bricks. Like that, millions of cells develop the body, and the cell have kind of same character. Each cell have same content of DNA, and the total content is called chromosome. Fun fact is, if you could, like if you see here, unwrap your chromosome. If you could unwrap the entire chromosome, you could start from this earth, you could go to sun, come back 600 times. That big is DNA content of a cell. So that means you could easily understand, without having any prior knowledge to biology, one dimension of DNA is definitely not in nanometer. It's really long. So that makes DNA is a one dimensional nanomaterial, right? A lot of people, when I used to talk with them, uh, as an expertise, I work on enzymes, I work on protein. So my research interest, my academic interest is majorly focused on proteins. So I always wonder why people don't take protein as nanomaterial. And when I started interacting with faculties who are working in the area of nanotechnology, in the interface of biotechnology, to my surprise, I have found that a lot of people think protein is really big material. So since protein comes under nanomaterial, I would like to know your opinion about that. So again, anyone who want to volunteer, what you think about protein's dimension? Do you think protein comes under nanomaterial or not? Anyone? No one? I'm sure my PhD students know about this, but I want to ask, specially put this question to the rest of the colleagues in this class, rest of my students, what you think. See, please interact, like without interaction, the class would be very boring. You might be wrong, that would not make any difference. Please put your perspectives. That will make the class more interesting. So anyone?
Yes, sir. That can be protein, can be nanomaterial, nanostructure, and nanoscale.
So put your name, please, huh? First,
Tejasman.
yes. So why you think protein comes under nanomaterial? What you think
Because
the width or the length of a protein?
uh, because that albumin, that's the product of proteins are mainly nano size. as far as my knowledge, sir.
Yeah. So, what do you think? How it correlate? You know the basic unit of protein, amino acids.
Yes, amino acid.
Yeah. So if if I ask you to give a estimate of the size, do you have that estimate, or you could
No,
say no, no problem.
that's I know about molecular weight, sir, but don't know about the size of amino
Fair
acid.
enough. Anyone else? Thank you, Tejasman. Anyone else?
Sir, Asmita here.
Asmita, please.
Sir, actually, I also know about the molecular size. Like, we measure usually protein in daltons.
Mm-hmm.
So, like, the protein size will depend on what type of protein it is and how many amino acids are there. So,
Right.
depending on that, we calculate the size of a protein. Like one amino acid is usually 110 Dalton, and then we calculate how many amino acids are there, and then multiply and find out what is the approximate size of the protein.
So if I say you have 200 amino acid, what would be the size of the protein?
That can be calculated under that's molecular weight only, sir.
No,
Tejasman here, right?
molecular weight only give you the weight, right?
Yes, sir. Yes, sir.
But we are talking about when you want to find it's a nanomaterial or not, you have to go through the size,
Length. Yeah.
the length, the width, the height, right?
Yes, sir. Yes. No idea, sir.
Yeah, and then I'm not surprised, but I'm at least happy that you guys start knowing the fact that protein is nanomaterial, because I will try to show you, and I will, when I do modeling, I will also give you the process so that in future you could easily calculate this. Okay.
Yes, sir.
So I start with a small protein which is called COM-EA, which is having a molecular weight of 12 kilodalton, and it is having amino acid 110. The size is 2.6 nanometer. Okay. So how the size is, because here by size we kind of like indicating volume, right? Because if you look at the protein, it's kind of globular, right?
Yes, sir.
Yeah. So how it's happened, because you wonder like how, first of all, when I will go to modeling, I will show you the mathematics of calculating the actual length. But the problem of protein is protein does not give you the allow you to measure it, because if you connect the chains of amino acids, you get the straight line, but then they fold back, which is called protein folding, a big enigma which I'm going to talk about in details. So, in that way, it is not possible to understand the volume. But here, nanopore, if you look at the dotted line in black, can you see in the slide?
Yes, sir.
Yes, sir. Yes, sir.
So this is a nanopore through which the protein is passed through, and so there is the apparatus the protein is passing through, and the nanopore is adjusted according to the size. So how it is adjusted, the final fit volume is calculated. Now you are understanding what is happening. So there is a instrument, let's say imagine it like a like cylinder of a glass. Okay, your protein is going from one point and passing it, and on the automated measurement place it could be adjusted, so it adjusts up to the protein would be fitted there, and this is there. So from there we get the size. Is it clear to you? A like good idea. We will go into much details though. So Molecular weight is 12 kilodalton. Amino acid number is 110. The size is 2.6 nanometer. So you could easily understand this is a 0D material. Agree with me, everyone? Anyone not understanding this? Anyone who don't understand, I want to repeat it but understanding these fundamentals would be very important for you to further, proceedings. Okay. So if someone didn't understand, I don't care how much time I will spend, but please stop me and ask me. Okay. I cannot go inside your mind. If you would be there in front of me, I would, look at your face and could understand. But because you guys are not here, please help me stopping me anytime. I would be the happiest if someone stopped me. Okay. Then coming to another protein which is called GFP or green fluorescence protein. This protein is 27 kilodalton, amino acid 245, again size is 4 nanometer, very well a zero-dimensional nanoparticle. Going to little bit bigger protein, this is like a protein which works on the development. This is a little bit of, I would say, not exactly globular protein with a molecular weight of 66 kilodalton, amino acid 600. The size is 5.4 nanometer. Proceeding to even bigger, this is IgG. This is immunoglobulin. Molecular weight 145 kilodalton. Size is 6.9 nanometer. 1318 amino acids are there. Then this is RNA polymerase, one of the biggest possible protein in the whole protein universe. 48 kilodalton, 4360 amino acid content, and the size is 10.5 nanometer. So I am giving you example of 5 different proteins. People who are out of the field, they are wondering, Sir is giving us example of proteins. What might be the average one, right? Do you guys have idea? What is the average molecular weight of a protein? Anyone? So this is a representation from protein data bank. Protein data bank is the bank where most of the protein, like all of the protein structures are deposited. And if you see here, average molecular weight is 300. If you look at here, it's the number of amino acids. So average length is 300 amino acid, which means around 330 kilodalton. 33 kilodalton. So you could easily understand that molecular weight of 145, molecular weight of 480 is way higher. And with those being way higher, still the volume is below 10 or around 10 nanometer. So in summary, maximum protein comes under the dimension of 5 nanometer. Then the biggest ones are within 10 to 15 nanometer. And that clearly says that protein is truly a zero-dimensional nanomaterial. Is it clear to everyone?
Yes, sir.
Yes, sir.
Now, coming to more detailing of biological macromolecules. you could see here different interaction of macromolecules, because interaction is very important. Here you see the green ones are DNA, the purple ones which are coming are protein, DNA protein interaction, protein protein interaction in the second picture, and the third picture represents the membrane, which also develop and take very important part in biological process. All the getting and all how a material goes from one side to next side, they are all organized or controlled by the membrane. So how to visualize biological macromolecules? If you see, started 1 millimeter to close to 100 nanometer around, all the tissue, cell, organelles comes into, and they could be seen by light microscope. Whereas the biological macromolecules, they could not be seen by light microscope, they could be seen by electron microscope. Okay. And in that point, again, I would like to compare between the field of nanotechnology and biotechnology, considering the biological macromolecules. If you remember, when we are talking about nanotechnology, we told the world of nanotechnology, the journey of nanotechnology was actually started with the innovation of scanning electron microscope, scanning tunneling electron microscope, transmission electron microscope, atomic force microscope. Similarly, the journey of biological macromolecules, which are in the range of nanometers, started with the innovation of protein crystallography, nuclear magnetic resonance, and very recent but another microscopy, cryo-electron microscopy. So basically, scanning electron microscope, transmission electron microscope, atomic force, cryo-electron, all are electron microscopy. The change of our innovation from using light as a source to visualize to electron as a source to visualize have changed the game forever. We will definitely give like knowledge about this. We'll discuss about this. But whenever you take a look, whenever you start thinking about the field, you see that the game changer, what is the game changer? The game changer is from the thought that light microscopy, where light is used as a source, changed to electron microscopy, where electron is used as a source. So this is one of the biggest change which brings nanotechnology or nanobiotechnology, the field what it is. So two things I would say is important from today's discussion. One is we talk about materials. We are talking about nanoparticles, nanomaterials, nanoscale materials. According to our conception, modeling of nanomaterial is not development of nanomaterial, is not building of nanomaterial. It is designing, it is modification of the existing material. Whereas in case of biomaterials, in case of biological macromolecules, rather than design, we first do development. We first start from the scratch, unite them, and develop into a complete model. So we will study both of them, but to know more about the biological macromolecule, we would study the macromolecules of life. We would try to answer the questions, what about the macromolecules of life? What are their functions? So who are the macromolecules? We already know that DNA, RNA, proteins. These are the three biological macromolecules of life. Are there anything else which could also join this? What are their function? Like, we know the macromolecules, but what are the specific function of them? How they grouped? Like I told, DNA and RNA both are nucleic acids. So is there anything else who are grouped? Is protein grouped with other things? How are they composed? What are their building blocks? How they are developed? This is very interesting information which will help us to do the modeling. Even how we could read them. So how to read them? Okay, you have a chain of molecule. How you start reading them? It's a very big field. It's a very beautiful enigma of life. And last but not the least, what about their 3D conformation? So remember, I told you take amino acid, you join amino acid, you get a stretch. But is that stretch defined life? Is that way they function as a stretch? Or there is other way they shape up into functional molecules? So from the assembling of the molecule to the functioning of the molecule, is it taking same form, same structure? We are going to look at them. I will start with basic introduction to the biological macromolecules. For the survival and growth, living organism require nutrients. Animal obtain nutrients by consuming food, while plants pull nutrients from soil. Animal, they consume food, and plant pull from soil. Many critical nutrients with polymeric nature are called biological macromolecules. The term macromolecule was first coined in the 1920 by Nobel laureate Hermann Staudinger. Staudinger was also the first to propose that many large biological macromolecules are built by covalently linking smaller biological molecules together. So, up to now, we know that there are biological macromolecules, but in this point, we are going to know that these macromolecules are actually developed from units. As we say, the cell develop the whole body, like smaller units from the larger biological macromolecules. So who are those units? How they come and chemically how they join and form would be very interesting to study. Biological macromolecules play a critical role in cell structure and function, and we will go further to know that there are four major classes of biological macromolecules. I told about classes. So one class is carbohydrate, and I have provided some popular pictures which will give you the clear idea of what are carbohydrates. Then another group called protein. We have already talked about proteins. Another group called fat, and another group we have already discussed called nucleic acid. There are two types of nucleic acids, ribonucleic acids or RNA. mRNA is one type of that. There are other two types, tRNA or transfer RNA, rRNA or ribosomal RNA. And there is DNA deoxyribonucleic acid. But this is the overall composition, this is the overall structure. We will go into the molecular level understanding, molecular level knowledge of all those four major classes of biological macromolecules. With that, I will finish here. From next class, in the next class, we will know about the details of this biological macromolecules. If you have any question, please ask.
Yes, sir. Sir, Ria here.
Yep.
Sir, actually, in one slide, there is one thing that proteins are zero-dimensional particle.
No, not zero-dimensional particle. Protein is zero-dimensional nanoparticle.
Yes, sir. Zero dimensional nanoparticles. Sir,
Yeah.
why called zero dimensional? That I can't
Yeah.
understand.
That you don't understand because you are not listening to the classification. Okay. In the last class, I'm taking you there. See, nanoparticles or nanospheres, nanoscale lengths are measured in all 3 dimensions. Do you understand that? If in all 3 dimensions the measurement is in nanometer, then it is called zero-dimensional nanoparticle. Is it clear now?
Yes, sir. Yes.
So,
Thank you, sir.
in case of protein, you see in any dimension, this is measured within 1 to 100, because now, like when we have already analyzed all, you know that basically all the proteins are within 10 nanometer in size, like in height, in width, and in length, right? So because it is less than 100 nanometer in all the dimensions, it is called zero-dimensional nanomaterial. Clear?
Yes. Yes, sir. Clear, clear.
Thank you,
Thank you,
Ria.
sir.
Anybody else?
Hello, sir. Good afternoon, sir.
Yep. What is your name?
Sir, Udit Narayan Sharma, sir.
Yeah. So Udit, when you
Sir,
ask
we
the question,
classified.
you say Udit and then ask the question. Yes. Continue, please.
Sir, we classify macromolecules and micromolecules in plant on the basis of absorption. But in animal, if we will talk about, then how? What are the basis to classify macromolecules and micromolecules?
Your voice is not clear. Can you repeat? I'm sorry. Somehow the voice is not coming clearly. Can you repeat?
Sir, yes, sir. If we are talking about plant, then plant we classify macromolecules and micromolecules on the basis of absorption. But if we will talk
No,
about animals,
I don't get it. In plant, we classify macromolecule and macromolecule and what do you say? I don't get it. Based on absorption.
on the basis of absorption, like a plant absorbs the macromolecules or micromolecules on the amount of absorption.
I
There is
think
a specific amount.
I think you are talking about macronutrient and micronutrient. Am I right?
Oh, yes, yes, yes. Sorry, sir. Sorry,
They
sir.
are two totally different things. Sudeep.
Yes, sir. I'm really sorry. Really sorry.
Yeah. No, you don't need to be sorry. See,
Yes,
we all make
I got
mistakes. We
confused.
all make mistakes, and I would be very happy. Whatever you have in mind, you ask. We come here to discuss. Please don't take it very seriously. Like today, probably by making a mistake, you help others to learn. Udit. So
Yes, sir.
nobody, you don't need to be ashamed. We are here to learn. After this class, I would expect you have all the basic knowledge, and that's it. I would not mind any questions. Even if I don't know, I don't mind. I have the courage to say I don't know, and I will come next day studying even harder. Okay.
Thank you, sir. Yes, sir.
To everyone,
Sir,
ask anything, any question. You find it silly, probably it might not be silly. Okay.
okay, sir.
Anything
So my
else? Any
question is
questions? Yeah.
how we classify macronutrients and micronutrients in animals. What are the bases?
Sorry.
If we'll
Sorry.
talk about animals, then what are the bases or what are the parameters on which we define macromolecules and micromolecules?
Macromolecule and there is no in that way. So macromolecules are molecules which are mostly polymer. In the next class, when we will go into detail, you will understand what we call or why we call them macromolecules. After knowing about them, but biology is a very interesting subject. It's a very layered subject. So each of, as you already pointed out something, like when we say micronutrient and macronutrient, when we say micromolecule, for one division, for one categorization, we have one logic. We go to the second, we come to the different logic. But yes, there are logics on individual classifications. So yes, coming to class will make things clear for you what biological macromolecules are, what their function, how they call macromolecule, why they call macromolecule, what are their subdivisions, how are they coming together, what are their chemistry. I hope this will be clear in coming 2 classes. Okay.
Okay, sir. Thank you, sir. Thank you so much.
Yeah. Anybody else? Okay. Thank you, guys. Yeah. Yep.
Sir, Asmita.
Yeah,
Sir,
Smita.
Asmita here.
Yeah.
Sir, the questions that you asked to write and send, should we send it before the next class?
No, you have to present it. Thank you for asking.
No, sir, not the second one. The first, you showed a set of questions.
Yes.
Okay, sir. Thank you, sir.
Yeah. See, again, my goal is just to involve all of you, because this is kind of a interesting and new subject. This will help you in a huge way to like choose your future research field also. So I want you to be involved here. Marks and all, they're important, but learning this course, I promise you will see that would help you in future. Okay.
Okay, sir.
Yep.
Okay, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thanks, everyone.